Welcome to PacBio's workshop at ASHG 2021. I'm Jennifer Stone, Vice President of Segment Marketing at PacBio, and today it's my great pleasure to share with you a presentation entitled HiFi Sequencing. See what you've been missing. HiFi sequencing, or HiFi reads, are the cornerstone of PacBio's technology. On our Sequel IIe platform, you're able to access long, up to 15 to 20 kb long, and highly accurate, 99.9% accurate sequencing reads. This combination of long and highly accurate sequencing has made PacBio the gold standard for de novo genome assembly across species. For human applications, we're seeing significant adoption of HiFi sequencing for variant detection in translational research, either with a whole genome or targeted approach. And there's a ton of excitement for the use of HiFi RNA sequencing. HiFi allows for full-length isoform detection, and researchers are finding that either in bulk or at the single-cell level, this is providing a new window into biology that's simply not visible with short-read technology. And lastly, I'd like to mention epigenetics. In general, our understanding of the impact of epigenetic modifications on phenotypes of interest is in its infancy. There's been some fantastic work published earlier this year that is really starting to show the power of gathering epigenomic signals straight off the native sequencing run. Getting methylation along with ACs, Ts, and Gs with no need for bisulfite conversion is an approach that will surely usher in a new wave of discovery. Touching on accuracy a bit more, I'd like to share a very detailed publication from the Association of Biomolecular Resource Facilities, or ABRF, that was recently published in Nature Biotechnology. The association compared all the different sequencing platforms, including the sequencing systems from Illumina, IonTorrent, PacBio, ONT, BGI, and Genapsys. The authors observed that PacBio had the lowest error rate out of all technologies. In addition, PacBio HiFi had the highest mapping rate, lowest non-mapping rate, i.e. the highest proportion of useful data, and even in challenging regions like homopolymers and short tandem repeats, or STRs. Significantly, because of this superior accuracy, the authors found that PacBio HiFi achieved the highest precision in calling clinically relevant variants compared to both short and other long-read technologies. Now let's put these long, highly accurate reads to work. Well, it all starts with a reference. What is the baseline that we compare things to? Many of us remember the publication of the first draft of the Human Reference Genome in 2001, and the completion of that reference in 2003. Tremendous accomplishments that have been the foundation, now 20 years on, for what is now the field of human genetics and the driver of precision medicine. But as we all know, the Human Reference Genome was not truly complete. It's taken the efforts of the Telomere to Telomere Consortium the last few years to finally and officially complete the Human Reference Genome. The preprint for this work was published just a few weeks ago and marks a major milestone for the field. PacBio HiFi sequencing was essential for the completion of this work, in that it provided the foundational technology from which the assembly was built and onto which data from other technologies was added. As the authors describe in the preprint, they leveraged the combined accuracy and length of HiFi reads to enable the assembly of highly repetitive regions, even centromeres, and resolve closely related sequences, such as segmental duplications. The work highlights the utility of HiFi reads to provide the most complete view of human genomes, finally resolving regions that have been impossible to see for many years. In fact, the authors summarize their findings, suggesting that at least 8% of the genome was previously overlooked and is now officially part of the reference, made accessible now with the long, highly accurate reads of HiFi. Beyond the completion of the Human Reference Genome by the Telomere to Telomere Consortium and the mapping of an additional 8% of the genome that was previously overlooked. PacBio is enabling discovery on multiple other fronts as well. First, I'd like to highlight the great work coming out of Dennis Lo's lab at the Chinese University of Hong Kong earlier this year. The team developed a methodology to directly examine 5-methyl-C at the whole genome level using the kinetic information from real-time HiFi sequencing, without the need for bisulfite or enzymatic conversions. In the PNAS paper, Professor Lo et al. Described the development of algorithms that leverage HiFi sequencing information from both DNA strands to improve the detection of 5-methyl-C. The paper describes its application to human whole genome sequencing and the benefits of characterizing the epigenome with long, accurate reads. It allows epigenetic phasing and allelic differential methylation. So just like the phasing of genetic variants, the ability to detect the methylation status genome-wide with long HiFi reads also allows for long-range epigenetic phasing information. Second, Trotsky et al. Out of the University of Queensland recently published a great example of the power of HiFi to resolve aspects of the human genome that were inaccessible before. The authors used deep, full-length PacBio cDNA sequencing of normal human tissues and cancer cell lines to hunt for pseudogenes. The authors described many novel transcribed pseudogenes that were not found in GenCode, and they further show that these are expressed in tissue-specific patterns. To assess the biological impact of non-coding pseudogenes, they CRISPR-Cas9 deleted a nucleus-enriched pseudogene and observed hundreds of perturbed genes. This study powerfully highlights pseudogenes as a complex and dynamic component of the human transcriptional landscape, only accessible with PacBio HiFi reads. And lastly, on this slide, I'd like to highlight a couple of exciting papers leveraging long-read RNA-seq or iso-seq. For example, in this cool new preprint, Barbara Wald and Ali Mortazavi used single-cell RNA-seq, single-cell iso-seq, and bulk iso-seq and ATAC-seq to make discoveries underlying the genomic basis of differential RNA isoform expression during differentiation. Here, from myoblasts to myotubes. They discovered a new method called long-read split-seq, which allows for an understanding of how transcriptional start sites change through complex biological processes, such as cell differentiation. In the second example for iso-seq, this time in the area of cancer research, a group out of China recently published a paper establishing single-cell survey of the cellular hierarchy in acute myeloid leukemia. Again, using full-length transcripts and cellular barcodes, the different mutational profiles in these heterogeneous cancer types can be resolved fully with HiFi iso-seq reads and assigned to the different cell types and clonal lineages. But just as some researchers are using PacBio HiFi to push into the unknown, making new discoveries of basic biology and cellular processes, others are pushing on previous discoveries to bring them closer to the clinic. The first publication I'd like to highlight uses PacBio to resolve the variation in human-specific tandem repeats that are associated with Lou Gehrig's disease, or ALS. Here, the authors used a targeted approach on PacBio to fully characterize the variation in length of the tandem repeat and its association to disease and its pattern of frequencies across human populations. This further enhanced our knowledge of the role of the WDR7 locus in ALS etiology. Second, I'd like to highlight a recent paper about a Chinese infant with sudden cardiac failure. The researchers studying this patient had identified 2 mutations in the same gene with Sanger sequencing. However, it was not possible to determine whether these were in cis or trans. Using PacBio sequencing, which allowed for single molecule reads of over 8 kb, the researchers were able to determine that the 2 mutations were in trans, the first one coming from the mother, and the second one was a de novo mutation inherited on the paternal allele. The authors note in their commentary that confirming the phasing was instrumental in allowing clarification of the genetic etiology and was helpful in counseling the patient and the patient management. And thirdly, another example of the power of long-range phasing is in the area of cancer research. As described in this science paper, led by a team out of Memorial Sloan Kettering Cancer Center in New York City, researchers found that multiple mutations in the PIK3CA gene are common in breast cancers. And of those, 95% are double mutations. These mutations are very far apart, or can be very far apart, and are difficult to phase with short read or even Sanger sequencing. But what they did find were that double mutations in cis, meaning on the same chromosome, those patients showed a phenotype of enhanced oncogenicity and greater sensitivity to PI3 kinase alpha inhibitors. So I've shared a few vignettes of how PacBio sequencing is making an impact in human genetics, but this is just the tip of the iceberg. Human genetics researchers are using HiFi as a new way of looking at human genomes, seeing regions for the very first time or in a more complete context, and thereby allowing for new insights and discoveries in many areas. Additional areas for impact is in the translational space include bringing HiFi sequencing to bear in rare disease, in pharmacogenomic studies, neurological diseases, cancers, reproductive health, and transplants. All of these bring exciting opportunities with unmet needs that can be addressed with PacBio HiFi sequencing. Now, shifting a bit to PacBio as an organization, we've had a very, very busy year. Kicking off 2021 with the announcement of our collaboration with Invitae to accelerate long-read whole genome sequencing into routine clinical care, and followed by a substantial investment by SoftBank into our company. We've announced a number of collaborations over the year with world-leading organizations such as LabCorp, Children's Mercy Hospital, and Rady Children's Hospital. And we've had a number of exciting product launches and acquisitions, namely Circulomics, and to extend our capabilities in extraction and sample prep, and Omniome, a short-read sequencing technology. A few additional words on Omniome. So first and foremost, we are so excited to become the first company to offer both long-read and short-read technologies, that we will be able to uniquely support customers with the right combination of sequencing technologies to address their needs. SBB, which is the chemistry supporting the Omniome technology, has exceptionally high accuracy, and that yields a lower limit of detection potentially. We are optimistic and really, really excited about this technology to go after applications that currently require very, very high depth or high coverage to execute, looking for a needle in a haystack. The higher accuracy will make those applications that much more accessible. We also see ourselves as being the best partners in the industry. That's one of our driving goals, to put the customer at the center of everything that we do. We're here to support you, to support your goals, your mission, and what you're really trying to accomplish, either in your research or the applied use of our technology. And we want to be your partner in helping you to do that. And then lastly, as one organization, PacBio with Omniome, there are tremendous synergies that we expect to be able to capitalize on, bringing the relative expertise of both organizations to bear in product development and how we come to market. Now back to the PacBio side and the long-read technology, we have a couple of teasers that we'd like to share here for new products that we hope to launch in the coming weeks. The first one is for a scalable whole genome sequencing workflow that's going to be a faster, simpler library prep, reducing time from about 8 hours today down to about 3 hours, bringing bead-based size selection and reduced DNA input for all kinds of sequencing. This will be a kitted workflow with an expected launch later this quarter. And then the second that we'd like to announce is a simultaneous detection of epigenetic information. We currently have research grade tools that are available to collaborators to explore the extraction of epigenetic signatures, methylation information from the sequencing runs off of a Sequel IIe. And we are in the process of productizing that offering and hope to bring it to market in the first half of next year. Now, shifting to ASHG, we have a packed agenda for this virtual conference, and I'd like to highlight a few of the ways that PacBio is participating and where you can go for more information on our products and what we're doing as an organization. So the first on the far left side is a fireside chat. I will be hosting Christian Henry and Richard Shen, Christian Henry, CEO of PacBio, and Richard Shen, outgoing CEO of Omniome, now part of the PacBio organization, in a discussion about how they see the future of genomics and the future of sequencing technologies. In the center, we have a list of all of our poster presentations, as well as one of the platform presentations that our scientists was fortunate enough to receive. So lots of really exciting and much more in-depth detail about our products and workflows in those sessions. And then lastly, on the right-hand side of this slide, the collab theater talks and the lightning talks being presented by a number of our marketing and technical experts across the company, focusing on things such as pharmacogenomics sequencing, methylation sequencing, and our new COVID genome sequencing kit that will be launching in a couple weeks. We have three very, very exciting seminar speakers. The first, Emily Farrow, coming from Children's Mercy Hospital in Kansas City. She's going to speak to us about the use of PacBio whole genome sequencing in increasing our understanding of rare disease patients and yielding new insights into the disorders of those children. The second talk that I'd like to highlight is a presentation on MassSeq, which is a highly multiplexed concatenated form of IsoSeq. And this is going to be presented by Aziz Al-Kafaji, pardon me, out of the Broad Institute. Really, really exciting work going on there. And last, but certainly not least, Dr. Henning Holstegge from the Medical University of Amsterdam, and she will be presenting on her use of whole genome sequencing on PacBio to study Alzheimer's disease. I'll leave you with our mission statement, PacBio, enabling the promise of genomics to better human health. We create the world's most advanced sequencing technologies. So excited to be here at ASHG 2021. Thank you for your attention, and we look forward to seeing you at our other events throughout the conference.